So a very warm welcome. It's great to have you all here, and we are very much pleased to have Kai Rothkamm here with the beautiful skyline of Hamburg. Kai is a trained physicist turned radiation biologist. He went through many stations from London to Oxford and ended up for now in Hamburg. And we met at a cancer meeting close by. So I'm very pleased to have a speaker here from the north to forge our northern cooperation here. And Kai's research is a lot based on what does radiation actually do to us? What does radiation actually do to the DNA? And I found it very interesting what he was telling me in a private discussion. So I'm very pleased that you're here tonight to speak to us. Everyone, please, you can ask many questions at the end. And without further ado, Kai, the stage is yours. Thank you so much for being with us.
Thanks very much for this invitation. Thanks for giving me the opportunity. And it all goes back to that meeting we had 2 years ago or so in Bad Segeberg, where I mostly discussed with you my former job, which was at part of the Department of Health in the UK. And I was based in Harwell in South Oxfordshire. And I was responsible for looking after radiation casualties, so people who got exposed to radiation in incidents or accidents. And we did biodosimetry for that. So I will cover that a little bit, but my topic for the last 9 years has really more been the analysis of and utilization of radiation biomarkers in cancer therapy, which will be the second part of the talk. And yes, for a start, radiation, I'm talking about ionizing radiation here, so nothing about UV radiation, although that is a major concern in terms of skin cancer, obviously. For ionizing radiation, the interesting feature of ionizing radiation is that it introduces ionizations, and they occur in clusters along the particle tracks. So for X-rays and gamma rays and so on, it's the secondary electrons which result from the initial ionization, which cause the clusters of ionizations, especially in the low energy range, so the track-end electrons that you can see here in the middle picture, which will induce a number of ionizations over a few nanometers of range. And if that happens to occur within a DNA molecule, then you will get multiple sites within a few nanometers of DNA that are damaged. And quite often this results in both of the strands of the DNA being broken. And that introduces a DNA double-strand break, which is regarded to be the most toxic and most severe lesion in the DNA that is caused by ionizing radiation. Radiation damages all kinds of biomolecules and other molecules, but typically for a cell, it doesn't really matter whether a protein is damaged thanks to radiation or a membrane has received some damage. It's really the DNA that is crucial and that is required to stay intact for the cell to survive. So double-strand breaks are therefore regarded as the most critical lesion induced by ionizing radiation. For chemicals, we typically have isolated DNA damage, not clusters of DNA damage, and therefore very rarely the occurrence of double-strand breaks caused by chemical agents. If you look at different radiation types, for example, high LET or densely ionizing radiation like alpha particles, then you will see even higher clustering of ionizations, causing even more severe damage to individual distinct sites in the nucleus, in the DNA, and that will produce lots of fragments of DNA, which are incredibly difficult then to put back together again to faithfully restore the original sequence. It's almost impossible. About 25 years ago, a Greek colleague who worked at the NIH, Amy Rogakow, he discovered a histone variant that is phosphorylated whenever one such double-strand break occurs on a chromosome. So on this C-terminus, position serine 139, there occurs a phosphorylation whenever a double-strand break is somewhere in the vicinity. And it occurs on hundreds of these histone variants that are close to such a double-strand break on a chromosome. An antibody was generated for this phospho-specific form of H2AX, and that allows individual double-strand breaks to be visualized in cells. And that has really revolutionized the research into double-strand breaks and their repair. And here are some examples how this can be applied. So this gamma H2AX staining that you can see here in the upper left corner shows you a distinct punctate pattern. So lots of, well, we call them foci appear after irradiation. This is after quite a high dose, 2 gray, typical dose you would give in 1/fraction of radiotherapy. If you take the cells 30 minutes later and fix them, stain them with this antibody and put them under a fluorescence microscope, you will see these foci appearing in the area where the DNA found here with the counter staining with DAPI. You can then, for example, look at other proteins and see whether they co-localize with these regions where double-strand breaks have occurred. This is one example of 53 binding protein 1, which is indeed co-localizing very nicely with the pattern that you see for gamma H2AX. Or another example here for MDC1, which is another repair protein. It's involved in the repair and the processing of double-strand breaks. And again, you will see nice correlation between these markers. After a longer time, so this was 30 minutes after irradiation, 24 hours after radiation, most of these foci have been resolved. They're lost. We assume, because repair has occurred, at least the kinetics with which this loss occurs is consistent with the kinetics of double-strand break repair assays that we use for physical measurements of the presence of breaks. So we assume that it correlates with repair events, and only a few breaks are left. And we think it's these few remaining breaks after a day which are really critical for cell survival. So a cell can typically handle most of the breaks, can repair them and survive, but a few may be left. And if they persist for several days, at any new approach through the cell cycle, any attempt to go through the cell cycle, to go through mitosis, the cell is likely to die. You can also nowadays use fluorescent tagged proteins like 53BP1 tagged to EGFP, as shown here on the upper right, and monitor directly the dynamics, the resolution, the occurrence of these foci in live cell imaging, as is shown here in a sort of time series. So nice opportunities to do some molecular imaging of the processing of these critical lesions. If you use chemicals like gemcitabine, for example, a chemotherapy drug, this chemical typically induces lots of DNA damage during S phase, resulting in what is called a replicative catastrophe. So the cell will essentially stop progressing through S phase, and it will more or less, the replication machinery will collapse under the burden of such a lot of damage. And then you will see these cell nuclei lighting up with a very strong gamma H2AX positive signal, but it's pan-nuclear staining. So you don't see these foci, but actually a very homogeneous, very positive staining for these gamma H2AX markers. You can also apply that to blood cells. That's shown at the bottom. And again, it's quite easy then to score these individual foci. Which allows you to, for example, take samples, blood samples from people who have been exposed to radiation. You can also use it in sections, FFPE sections, so paraffin sections of tumors. Here is an example for a xilograph tumor, where the tumor had been irradiated with 2 gray, and then we looked at foci at 1 hour, 4 hours, 24 hours. And again, you see that these are resolved over time. You can frozen sections and do the analysis in different organs and tissues, as you can also see here at the bottom. So a nice tool to do in situ visualization and also quantification of DNA damage, double-strand breaks, which are induced by radiation in different organs and tissues. When you look at the dose-response relationship for these, it turns out that this is clearly linear. That's consistent with previous physical assays, such as pulse field electrophoresis assays, where you simply measure the total or the average molecular weight of your chromosomal DNA. And obviously, the more breaks are introduced, the smaller the molecular weight on average. In order to do that, you need to resolve the chromosomal DNA using pulse field gel electrophoresis into fragments of megabase pair sizes. And the quantification of that is quite consistent with the number of foci that we see per gray that are induced by radiation. So you essentially end up on the more or less on the same axis. But the nice thing is with these pulse field measurements or other physical assays for double-strand breaks, we were restricted to doses of 10 gray and more, and it only worked really reliably at 20, 30, 50, 80 gray, which obviously is not very physiological. It's not interesting in terms of radiation protection, and it's severely limiting because you don't know whether you completely overwhelm the cellular machinery for repair of damage using such supra-lethal doses. With this foci approach, we were able to decrease doses down to, well, a milligray or a couple of milligray, and still get reasonably reliable data, depending a bit on the background of so the background noise of the cells you're looking at. But there are cell systems like blood lymphocytes or stationary primary human fibroblasts, where you can indeed measure the effects of radiation exposures on the order of a few milligray using this assay, which was really a major step forward. And that obviously opens up a fantastic way to analyze samples from people who have been exposed to low doses, and I'll come to an example later. Up to then, and for the last 60 years, the gold standard for assessing doses in people who were exposed to radiation is the dicentric assay, where you look at chromosome aberration formation. So essentially, you look at the chromosomes in blood lymphocytes, and when a double-strand break has occurred in one chromosome, quite close to another break in another chromosome, then occasionally these break ends will be mixed up by the repair machinery, resulting in such structural aberrations, as you can see here on the right. So you will end up with a chromosome containing 2 centromeres, called the dicentric chromosome, and an acentric fragment, which contains no centromere. And these acentric fragments are then lost from the daughter cell nuclei after cell division, and they end up in the cytoplasm. And the cell is no longer able to use the genes that are encoded on these acentric fragments. And if one of those is essential, then the cell will eventually die from because it has lost that essential genetic information in the nucleus. These aberrations occur in a linear quadratic manner, as you can see here. So there is a linear component at very low doses, and that is essentially all the events that occur within one radiation particle track. So if you have one electron track passing through 2 chromosomes and causing a double-strand break in each of these, then the interaction between these breaks will give rise to a double-strand to a chromosome aberration, to a dicentric chromosome, but that happens quite rarely. So it's not a very frequent event for sparsely ionizing radiation. So it's different for alpha particles, where you get that quite a lot. But at high doses, you have quadratic interaction, because you can have breaks from separate radiation tracks being close enough to interact. And obviously at high doses, you'll have thousands of radiation tracks in a cell nucleus. Roughly, you will get, well, one electron will deliver about a milligray on electron track in the cell nucleus. So at 1 gray, you will have 1,000 electron tracks, and it becomes quite reasonably realistic that at 2 double-strand breaks induced by different electron tracks will actually be close enough to interact, and then you get a quadratic component, which is added to that linear component. So that explains this linear quadratic behavior. And with that information, if you know how many aberrations you get per absorbed dose, you can actually do biological dosimetry. So you can find out if you have a person and find that in that person's blood lymphocytes, there's on average 1 aberration per cell, then that would correspond to about, well, somewhere between 4 and 5 gray total body exposure. And that's the basis for the gold standard biological dosimetry that has been used for the last 60 years, and that's still the gold standard. And for foci, we realized we could essentially do the same thing, but we would be able to do that at low doses, because as you can see here, for the chromosome aberrations, it's extremely difficult to see any excess aberrations at very low doses. So it's really limited to doses of 100 milligray X-rays, or similarly sparsely ionizing radiation, or higher. So 100 milligray is really the lower limit of anything that's possible. And for radiation protection, we are interested in tens of milligrays or even lower doses. And for that, the assay is completely blind. So with this foci assay, we realized we can actually go down to a few milligrays. So that opens up new opportunities. And for example, we can add that to the portfolio of dose assessments method for accident scenarios. And there are essentially 3 groups of assessment methods. There's physical dosimetry. In an ideal case, every person will have a dosimetry badge, which could be a sort of OSL or film badge or something like that. And you would have established methods for very sensitive dosimetry of those badges. Occasionally, the person will have the badge somewhere on their work coat and leave the work coat hanging somewhere and go on holiday. And then 3 weeks later, the badge will show something. And nobody knows whether the person has actually been exposed or whether it's just that the coat with the badge was placed in the wrong place and got exposed. In those situations, you need another method to confirm that this person was actually exposed or not. And you could use some fortuitous methods. For example, you could do luminescence assays for other materials. So if a person has some, shirt buttons, and the exposure must have happened very recently, you could actually look at the luminescence signal from that shirt button, but it fades extremely quickly. So it's not a very reliable marker if you don't know exactly when the irradiation happened. Or you could use a tooth or extract do a microbiopsy of tooth enamel and then analyze that using electron spin resonance. So that would be another fortuitous approach where you don't rely on having given a person a badge in the first place. And there are some bioassays which are interesting if you think somebody has been exposed or has had an internal intake, so has had inhalation, incorporation, ingestion of radioactivity. But all this is complemented by biological assays, mainly the gold standard dicentric assay, the cytogenetics. There is an assay based on fish, so that fluorescence in situ hybridization, which utilizes essentially chromosome painting. So each chromosome gets colored in a different color using probes, hybridization probes, and then you can detect also translocations, not just these dicentric chromosomes, but also balanced translocations, which in a normal DNA staining would look exactly the same, whether you wouldn't see the translocation. And you can do some other fancy things like premature chromosome condensation, or wait another day for cells to divide to go into cytokinesis, then you can look at micronuclei formation, which are a consequence of these acentrics which form during the misrepair of breaks, which are accompanied by the dicentric. But it's an easier assay to do, but takes a bit longer until you get a result. So in here, we thought, yes, gamma HTX foci might actually be a nice contribution, might be a nice addition. There are some other experimental markers which are being tested and have been tested for many years now, some biochemical markers of exposure, which work fine in really critical settings where you have sort of the question, is that lethal or not? So at 6 or 8 gray total body exposure, you will get some markers which can be useful. There are some mutation assays, and increasingly people are looking at gene or microRNA signatures, which will reflect the exposure level. And certainly you will see an increase there, but it's quite hard to find a robust dose-response relationship for these gene expression-based assays. The third column for this dose assessment testing is clinical dosimetry, where you would look at effects like vomiting, diarrhea, blood cell count changes, or skin reactions, which are all reflective of an acute radiation exposure. But of course, they are not terribly specific. If you tell somebody they may have been exposed to a high dose of radiation, it's quite likely that just for psychological reasons, they may feel unwell and may start vomiting as well. We've had that. So it's not the best marker to use as an exclusive marker. So in a NATO research task group, I don't know, 10 years, 15 years ago, we decided that we needed to test some of these assays to see whether they can be used in an emergency type approach. So triage, just identify who has really been exposed to a significant dose and who hasn't. And there we compared the gamma HTX assay, dicentric assay, micronucleus assay, but in addition also gene expression and I think premature chromosome condensation as well. But our lab, so the lab that I was running in Oxford at the time, it was involved in the H2X, DCA, so dicentric and micronucleus assays. And we were sent 10 samples, had no idea what they were, and analyzed them. And as you can see here, in a quick and dirty approach, so we had to use the assays as quickly as possible because we needed to deliver the information as quickly as possible. We were able to deliver the foci results within a few hours, I think 7 hours for all 10 individuals after receiving the blood samples, whereas it took 3 days, simply because you need to stimulate lymphocytes and then wait for them to enter the cell cycle, go through S phase, go into mitosis, and there they are stopped, and there you can fix them and then analyze them, and it takes ages to just score all those metaphases, all these chromosomes, which are then spread out, and find a few aberrant chromosomes, which will then be able to give you an answer in terms of a dose estimate. And even longer, it takes for the micronucleus assay another day, because then the cells have to move on through mitosis into cytokinesis, and it takes 4 days essentially for them to arrive there and then to be tested and measured. So in terms of speed, clearly this foci assay was far superior, and it worked also quite well in terms of the accuracy. The precision, so if you look at the mean absolute difference in the dose in gray, it was as good or as bad as the dicentric assay, so just under half a gray deviation on average, which is a lot, but as I say, this is a triage assay where you want to know, has somebody received more than 2 gray or more than 4 gray, and then you know roughly whether you can send them home, send them to hospital for monitoring, or send them for very urgent clinical assessment to see whether they may suffer from bone marrow failure. And the problem is that gamma HTX assay is extremely time sensitive. So if you receive a sample and you don't know exactly when the person was exposed, then you are completely in the dark with that assay. And the longer you have of a time gap between the time of exposure and the actual time the sample is taken from that person and then fixed, the longer that gap, the less accurate the assay becomes, because you will have less signal left, because you're looking with gamma-HTX, you're looking at unrepaired double-strand breaks, and the repair rate is on the order of hours, the half time. Whereas for dicentric chromosome formation, they form in with the same half time as the repair events, but then they are stable for weeks and months until you have turnover of lymphocytes, and then they get lost. So after months, they will decline. The same is true for the macronucleus assay. So none of these assays are great if you want to know whether somebody was exposed in the 1950s, which is another application where we look at, for example, people who worked on the Soviet nuclear bomb development in the Mayak plant in the southern Urals. And they had high exposures in the 1950s, and the survivors have developed, obviously, health complications. And now well, as a scientific community, we needed to reconstruct the doses for those people over the past 10, 20 years. And for that, none of these assays are great. But this translocation assay, for example, it's fantastic. That's a fantastic assay for lifetime cumulative dose assessment. So very different applications necessary for different scenarios. And the gamma-HTX assay, unfortunately, only works fine if you get your sample within a day or so. And you need to know when exactly the exposure happened. But clearly, there are situations where this works rather nicely, especially if you know when the exposure is going to happen, such as in a set of patients who receive CT scans. So that was something we tried as a first biodosimetry attempt with this method, where we, well, there was an appointment for each irradiation, so we could turn up within minutes after the exposure and take the first blood samples, and we repeated that for, well, the first half hour, and then fixed the lymphocytes and stained them for gamma-HTX and counted the foci. And indeed, we could observe an increase. We even had the luxury of being able to take a blood sample before the CT scan. So we had a baseline level as well. And in such an ideal setting, you can actually resolve doses down to several milligray, as you can see here. So this is essentially patients receiving a chest, abdomen, pelvis CT scan in red. Or we had one patient who only got a chest CT scan in orange. And we compared that with in vitro exposures to 20 milligray, just as a sort of ballpark figure to give us a reference value. And indeed, the chest, abdomen, pelvis scans were perhaps slightly lower than the 20 milligray in vitro exposure. So that makes sense. And if you know, we developed a calibration curve for that, a reference curve, and then could calculate back the doses, which were, as shown here, between 13, 15, 20 milligray for each of these exposures. And that was consistent with the Monte Carlo simulations that were done during the quality assurance procedures for that CT scanner. So we were quite happy. So this is the IM here. The black bar shows what the Monte Carlo simulation gave us in terms of a dose estimate for the blood, which is a bit tricky because normally you don't get the data for blood. Other organs are available, but for blood we had to do some tricks. But clearly for the chest CT scan it was lower, and we were still able to distinguish that from the baseline, which was rather nice. And as you can see, it can be really easy. If you see these individual dots, they are very easy to score. Sometimes it doesn't look as pretty as this. And at that time, we did all that manually, so scoring by eye on blinded samples. Nowadays, we are increasingly working with software, which gets better and better by the day. And there are now the first neuronal network-based machine learning approaches for scoring these signals, and that, well, makes certainly our students happy because they don't have to spend day and night looking through eyepieces and counting these foci. There was a stage when I actually could see foci when closing my eyes, lying awake at night. Yeah, not very recommended. Okay, what we also realized is that, well, obviously different people show different amounts of signals. So the number of foci, especially the longer the time progressed after exposure, the number of foci varied increasingly between individuals. And so we thought, as we know that double-strand break repair capacity is a very important determinant for radiosensitivity. So if you can't repair the breaks quickly, then the cell is going to be more radiosensitive, is more likely to die from the same irradiation dose compared to another cell. We thought we could perhaps test whether that might also be true for patients, for individuals. So we got together with clinicians who had been involved in very large radiotherapy clinical trials. And those were for breast radiotherapy. Essentially, these were patients which were involved in the START A and START B trials in the 1990s and early 2000s in the UK, which was looking at hypofractionation versus normal fractionation for breast radiotherapy. And some of those patients had developed extremely severe late radiotherapy effects, so very severe hardening of the breast, fibrosis, shrinkage. We looked at the most severe 7 cases among over 1,000 patients and compared that with 7 controls, just to set the bar low for us to see whether that will indeed show us that these molecular markers for radiation damage that we were looking at, these foci and also the chromosome aberrations, whether they indeed have anything to do with a clinical response that was observed in those patients. And we were very happy to see that indeed for samples of blood that were irradiated irradiated with 4-gray and then incubated for 24 hours to allow for repair of the damage, there was quite a distinct difference between cases and controls. And there was no difference when we looked at immediate exposures, so just half an hour repair time after half a gray, that there was not much difference between those cases and controls. And indeed, we would have been able to, well, with a sort of 80% accuracy or so, to identify those patients which show an increased number of foci, and with these were also the severe cases, except for one case where we would have missed, I think, with that approach. So that worked really nicely. The same is true for the chromosome aberrations. Again, much higher level of total aberrations in the cases compared to the controls, and similar results for exchange type aberrations or for deletions. The totals are just a combination of the both of these. And again, even better separation of those cases from the controlled, where no late adverse effects of radiotherapy had been observed. But of course, this was a very artificial setting. So we were looking, as I said, at the most severe late effects, patients with the most severe effects. So they were probably among the most radiosensitive. The question was, can we actually apply this prospectively? Would we be able to predict whether a patient will respond more sensitively to the radiotherapy compared to the average patient? And for that, we did a study which involved patients from the FAST and the CHIP trials, which was for breast cancer, the FAST trial, again, hypofractionation trial, and for CHIP was for prostate cancer hypofractionation. And there we recruited in total just over 400 patients and looked at chromosome aberration formation, gamma HTX foci early on and residual, and we expected only the residual to give us any significant effect. And also at this micronucleus assay, which I had mentioned before. And in addition, a colleague from Montpellier, David Azria, he had worked on, yes, he's still working on another predictive assay for radiosensitivity of patients, which is called the radiation-induced lymphocyte apoptosis assay. So they're looking at apoptotic cell death in lymphocytes following irradiation of those lymphocytes ex vivo. So they are taken out of the patients, irradiated, and then I think 48 hours later, they analyze these lymphocytes for apoptosis. So we compared all these assays for those 400 blood samples with great hope, and got these results, which initially first showed us that, oh yes, we have 3 different scorers that we involved, and they did differ slightly in their scoring approach, but unfortunately we had no significant association at all with late effects for chromosome aberrations. The same is true for the gamma H2X assay. We saw no significant association with late adverse effect. And the same was true also for the micronucleus assay. So really disappointing. It turned out, because this was done more or less prospectively, so when we did the analysis, we had no idea who of these patients was going to develop any late effects. And thanks to improved technologies in radiation delivery, so IMRT had come along and so on, the rate of adverse effects was lower than it used to be in the past. So clearly the medical physicists had done their job very well, so that we ended up with, I think, only 3 or 4 cases among 109 breast cancer patients in the FAST trial, and there were 20, 30 with patients with severe late effect in the CHIP study, but we couldn't identify them. We had no hope. So clearly, these assays are not really useful if you want to apply them as a sort of predictive assay for to test patients for abnormal responses to radiotherapy. It doesn't work. For the lymphocytes, it sort of seems to work. So there we get, at least for the breast cancer patients, clearly an identification of, and these are patients who have a very high rate of apoptosis. And in this case, this actually means for these patients that they show a very efficient response to radiation. So the signaling is very active in these patients, and that seems to protect them from the late effects. So they showed a nice correlation with the occurrence of late effect. For the CHIP, it was much more mixed and not significant. I think we can skip that. Essentially, well, we as a byline, while we are doing it, we thought we can just as well do dose estimates for all those 400 chromosome aberration-based analyses. And that showed us that, we were roughly estimating the 6 gray correctly. Well, we were overestimating a little bit. We also got samples from 8 volunteers, because one question was, do all radiotherapy patients, are they actually comparable to, or cancer patients, are they comparable to healthy volunteers? And all these patients had previously received radiotherapy, so would that have changed their response? But the averages are very similar. So there's no effect, no difference between these cancer patients who had received radiotherapy when we then irradiated lymphocytes ex vivo, and when we irradiated lymphocytes ex vivo from 8 healthy volunteers. And we had some 8 occasions of late delivery of samples. So it took 2, 3, 4 days, I think, for those samples to arrive rather than just overnight, but that had no effect. So in that respect, the assay is very sensitive, very robust. Okay, we're switching subjects now, coming back to another project that I was involved in, and that was about synchrotron X-ray microbeam radiotherapy. So there, those colleagues, they were from Australia, from Monash University in Melbourne, and they had the problem that they were using these very fancy irradiation geometries where you deliver extremely high doses in pencil beams, which are only 30 micron wide, and then they have gaps of 200 micron between individual radiation tracks. And with that approach, it seems, at least in experimental systems, you can kill tumors quite efficiently, but protect the normal tissue. So this is it's called spatial fractionation, this approach. And it's still in an experimental stage, but at the time, what they needed was really a marker which they could use in the tissue to show them where the radiation tracks actually were in their block in their specimen. And for that, we collaborated. The second question was then to see, can we actually measure the amount of damage caused along the track or between, especially between the tracks, so in these valleys, dose valleys between neighboring tracks, because it's apparently the valley doses that are especially important for we think, for the biological effectiveness of this radiotherapy approach. And indeed, if you do gamma HTX staining of these xenograft tumors, of these murine tumor grafts, then you can visualize the radiation tracks simply by positively stained cell nuclei for gamma H2X, and you can measure it up, and it turns out, yes, that we get the 200 micrometer distance between each track to the next one. And surprisingly, so this was done in samples that were fixed 4 hours after irradiation. In these grafted tumors, if we waited 12 hours before processing them and fixing the specimens and then putting them into paraffin blocks, then this nice distinct pattern of the radiation tracks got lost, and got even more lost after 24 hours. So clearly, it shows us that at least in this tumor system, in this experimental tumor system, where tumor cells have been injected into the mouse and then left to grow a tumor, these tumors seem to be extremely mobile. So the cells seem to move around quite a lot, so that within hours they completely move their positions. And that's quite in contrast to what happens in normal tissue. So here we see skin, murine skin, stained for gamma HTX, and you can see at 4 hours, 12 hours, and 24 hours, always exactly the same pattern where the radiation hit the intact nuclei of the skin layers here. So there's no washing machine effect, as we called it, that we've seen in the tumor, but actually everything is preserved geometrically for at least 24 hours. And even at 48 hours, there was the signal got weaker, but there was not much movement of cells. Yeah, so for the second question, we again generated a reference, a sort of calibration curve to tell us how many foci we can count per unit dose, and then applied that to the samples that had received microbeam radiotherapy, and then measured the dose, or the number of foci on the right side, as a function of distance from the microbeam radiation therapy track. So we initially looked at cells very close, 20 to 40 microns from the nearest track, center of a track, and that gave us these dose distributions. And the further we moved away from the next radiation track, the lower the dose, as you would expect, because you will get less scattering of radiation. And from that, we could then calculate peak to valley ratios for the dose, which were then on the order of about 30 to 50, which shows us that 50 times more dose is given in the actual radiation tracks compared to the furthest area away from these tracks, which is about 80 to 100 micrometer. Which was, again, reasonably consistent with film dosimetry. So they used GAF chromic films, which were put essentially exactly on the in the same position and actually onto the mouse during the irradiation, and which also showed peak to valley ratios on the order of 30 to 50, depending on exactly the setup we were using. Okay, now we switch again, because now, this is what we've been doing since I moved to Hamburg, in our lab at UKE, where we get more and more interested in tumor cell radiosensitivity and in approaches for radiosensitizing tumors. But I've already spent 45 minutes, so I will not continue for too much longer. So here we're looking at using these markers as essentially a surrogate for radiosensitivity. As you can see here, it correlates nicely with surviving fractions, and therefore we can then use it as an early endpoint, early readout for radiosensitivity, and study the impact of these different repair pathways and how they contribute to tumor cell radioresistance, and how we can target them in order to achieve better therapeutic outcomes. But I think I've spent enough time and would just like to finish by thanking all the external collaborators. And most of what I've shown you was really done during my time in Northwood at Mount Vernon Hospital at the Gray Lab, and at what is now called the UK Health Security Agency, which used to be called Health Protection Agency or Public Health England. So they change their name every five years or so. These colleagues, Liz Ainsbury, Jane Mockett, Stephen Barnard, they helped a lot with all the biodosimetry work that we did. Yes, and with that, thanks a lot for your attention, and I'm happy to take any questions.
Thank you so much, Kai. That was very, very interesting. Very cool. Thank you. Are there any questions from the audience? I start off. Can you explain me again what exactly is this alpha H2AX doing?
It's a gamma H2AX. so H2AX
gamma.
There are histones in the so histones form the chromatin. They are part of the chromatin structure. They are the basic building blocks for nucleosomes. And there are different types of histones: H2A, H2B, H3, H4. For H2A, there are different variants. So it's a bit complicated, but essentially H2AX is one of those building blocks that are necessary to make DNA into chromatin. So DNA is wound around those nucleosomes, and these histones form the basis for these nucleosomes, and so they are intrinsically linked to the information on the DNA and to any processing, any metabolism that involves the DNA. They need to be pushed out of the way when you want to transcribe a gene, or when you want to duplicate the DNA. They need to be moved around, and they are also used for example, for silencing gene loci, but then they are methylated, and this specific variant is phosphorylated. So a phosphate group is added whenever there's a lesion, a DNA lesion in the vicinity. So within a few, 100 megabase pairs or so. So quite a large distance. And therefore you get hundreds of these histones getting phosphorylated after one break has occurred. And that allows you to just look under the microscope and see these foci, and so we just use them as a marker. They have a function, so they coordinate the or they allow the repair machinery to be recruited, and they often enough you need to open up the chromatin in order for the repair machinery to actually access the break site, and in all this you need obviously you need the histones to be moved away and so on. his H2AX has a function in that, but it also has a function in stopping the cell from progressing through the cell cycle. Because obviously, if there's damage, then the cell wants to repair the damage before it enters any critical cell cycle phases, such as S phase or mitosis.
Okay. And how do these H2AX know that there was a break?
Yes, so there's that's an interesting question. So there seems to be a sort of there seems to be a police patrol for DNA damage, which glides along the DNA all the time. And
other proteins.
So there are proteins which will, yes, and they will, as soon as they find a break, they bind, they recruit a kinase called ATM, ataxia telangiectasia mutated, which is the gene that is also responsible for this disease called ataxia telangiectasia, which occurs in people who are extremely radiosensitive. And this ATM kinase, a kinase is phosphorylates target proteins, and one of the main target proteins for ATM is H2AX. So that's how H2AX gets phosphorylated.
Another chance for the audience to speak up. Otherwise, I'll keep talking and asking. So a double-strand break, right? That basically kills the cell.
If it's not properly repaired, it does, yes. So if you have only one and you have an intact repair system, then the break ends will be recognized, they will be protected from any further degradation, and then will be realigned and repaired, and everything's fine. If you have many of these double-strand breaks in a small volume close by, then often enough, because the repair event is the repair pathway is not very accurate. It will take the next best ends and just realign them.
Stick them together.
So it doesn't care whether they fit or not, and whether they come from the original break or not, and thereby you often get rearrangements. You'll often have the situation that break ends from different chromosomes then get misrejoined to form a chromosome aberration. And then the cell has to deal with that, and that can kill the cell if you, for example, end up with a dicentric chromosome, because then you'll have two centromeres, and during the mitosis, during cell division, this dicentric chromosome will be pulled into both daughter nuclei, because both centromeres will be target for the spindle apparatus, and you can get stalled mitoses and cells die in there, or you can break the chromosome again mechanically by pulling it apart, and you have loss of these acentric fragments with the bits the other bits that got misrejoined, which have no centromere, and therefore they don't get passed on to the daughter nuclei, and they are left in the cytoplasm, and then they are not available for transcription, and therefore the genes, if there are any essential genes on them, then they will not be expressed, and the cell will run out of the protein pool for those essential genes after a couple of divisions, and will die.
So that basically kills cells, and that is for the entire organism. That may be harmless if you have enough of those cells.
Exactly, yes. So it's not a big problem if you don't have if not too many cells have suffered. And that's the problem with acute radiation syndrome. So acute radiation syndrome occurs when the function of an organ is no longer
given or something.
Yeah, upheld because too many cells are being killed or have been killed by the radiation. And the first or the most sensitive organ for that is the hematopoietic system, where you will have loss of white blood cells, and the patients then get extremely vulnerable to infections, and they die from the infections. The next one would be the gut, where again you have a high cell turnover, and if too many stem cells in the gut have been damaged, severely damaged, and then the walling of the gut and of the
lining goes off, right? Of the
yes, and thereby you will have then invasion of gut bacteria into the patient and sepsis, and yeah, again, they die. So that's at 8 to 10 gray. You would observe that. And that's why it's important to know, how much dose have people received, so that you can take precautions and isolate them, and perhaps give them growth factor to stimulate the regrowth of the target cell population in the organ that you need to rescue as quickly as possible, or even do bone marrow transplantation if the dose was really, really high and the whole body was affected.
Timo, I see your question. I'll just do one follow-up here. And that does not cause cancer, right? If the cells die.
If they die, no, that's perfect. They don't cause cancer. If they die in a very uncoordinated way, that will cause some inflammation. And inflammation can be a driver for cancer, obviously, but in general, no. It's the surviving cells that survive the radiation exposure, but that may have had a rearrangement which can be tolerated by a cell, such as these translocations that I mentioned. They can be translocations can be formed in stem cells, and they will be passed on to daughter cells without any problem. And that's how leukemia, for example, arises from the famous Philadelphia chromosome. And radiation introduces such translocated chromosomes. And then it depends on exactly where the breakpoint is and how critical the genes are at the breakpoint, whether one of them has been disrupted, which is a tumor suppressor gene, or whether an oncogene has been activated because a regulatory sequence is now put next to it thanks to the misrepair of the break.
Yeah, I mean, for
but
cancer,
it takes years.
usually.
So you have years for leukemia and decades for solid cancer until you will actually see a clinically manifest tumor. So and that makes research into the mechanisms obviously very hard, because funding agencies don't give you money for 30 years of research without a result. So
and that must be then by chance sort of a mutation or a translocation that will cause proliferation or something.
Yes, which will give the cell a growth advantage, and then there will be additional mutations will be necessary in order for the cell to become invasive or bypass other control mechanisms. So it needs several of these events in order to become a fully
malignant
tumor. Yeah. And we know that mostly from A-bomb survivor data, really, from Hiroshima and Nagasaki, because there we knew exactly when the exposure happened, and many people were exposed. And I think the whole radiation protection system is based on the health effect analysis of these survivors.
And that took really, really long, you said, right?
Well, they still survive. Some of them are still surviving, and we now just recently found out about the cardiovascular effects that occur even at relatively low doses of radiation, because they took, again, 40, 50 years to show up.
Wow. Yeah, I didn't know that. Timo.
Yeah, can you hear me?
Yeah, yes. This is our medical physics expert.
I'm one of them. It's very interesting how you go into the detail. Actually, I have a question regarding stochastic and deterministic cell damage, or damage for the patient, essentially, because you kind of motivated me, because you showed the diagram of the double-strand damage down to a milligray. And now it's a bit the question, when you talk about radiosensitive or not so radiosensitive patients, does it actually mean also that there's no threshold actually, and we should reconsider this, or do you think thresholds are still a valuable thing, and this radiosensitivity is just meant on the stochastic part, on the severeness of the stochastic part? I mean, can you kind of
yeah.
It's also easy if you read it in the books, but if you now have
paid
attention,
it gets very confusing very easily, I know.
Yeah.
So for the radiosensitive patients that we looked at, radiosensitivity was determined or was defined as the occurrence of late severe deterministic effects. So they are clearly not stochastic effects, so fibrosis or rectal dysfunction in the case of the prostate patients, or bladder dysfunction. No, they are deterministic effects of high dose radiation, clearly. Of course, radiosensitivity is also discussed as an important question for occupational exposures. Ideally, you wouldn't want to send radio-sensitive workers into a radiation environment. So if it were possible to find out whether somebody is hypersensitive to radiation and to the stochastic effects of radiation, which means that they would have an elevated cancer risk, because the cancer risk has no, from what we know, has no threshold. And from the mechanistic work we do, we realize that a single electron track can introduce such a misrepaired double-strand break, which would cause this rearrangement. So it makes sense that there's probably no threshold. Then it would be nice, only to send those workers in who have very well-functioning repair machinery, which will take care of those breaks more efficiently than perhaps a worker whose major primary repair pathway is sort of compromised and not fully operational. And that's not something you typically know. You find out during radiotherapy by chance, because those people who are radiosensitive because of such a molecular defect, they can live a normal life until you challenge them with extremely severe doses, such as in radiotherapy. But they then show, of course, the deterministic effects, because we don't typically, there are secondary cancers as well, but they occur decades later. The corporate memory of those patients isn't up to proper epidemiology. So our cancer hospital, I think, doesn't have data for patients who received radiotherapy 50 years ago, which would be interesting. We have been discussing with the Hamburg Krebsregister whether it should be possible to have a sort of more long-term archive of cancer and also radiation effects.
I'll read the question from the chat. In the synchrotron, Andreas Berg is asking, in the synchrotron microbeam irradiations, tumors seem to be more sensitive to such type of irradiation. The DSB results seem to follow the physical dose peak to value ratio. What radiobiological mechanism is then responsible for the different biological behavior of tumor and healthy tissue? Thank you.
That's a great question, and I think a lot of people have been asking that question for a long time now. There are some theories. So one is based on a so-called bystander effect. So we know that cells communicate with each other. We know that if you irradiate, and we also did that with microbeams in vitro, you can irradiate one cell and see what happens to the other cells in a dish. And you will actually, a few cells will die in the dish because you irradiated one cell. So this one cell will send out signals which will force others to react, and some of them decide to die. If that happens in a different way in tumors compared to normal tissues, then that might contribute to such an effect. Then there is the issue of blood vessels. So tumor blood vessels are much more chaotically organized, or not organized really, and might be more vulnerable than blood vessels in normal tissue. So if you use these huge doses, so typically they use several hundred gray peak dose. And we're talking in one go. We're talking 100, 200, 300 gray that are given as peak doses. If that such a radiation dose hits a blood vessel, hits the endothelial cells in a blood vessel, it might shut down the blood vessel. And revascularization might be more efficient in normal tissue than in the tumor. So it might be, so really killing this tumor by essentially starving it to death might be one mechanism that has been discussed. But so far the findings are quite inconclusive which of these it is, and there might be other effects that we haven't quite fully grasped yet. For example, there's the flash effect. You know, in microbeam radiotherapy, the whole dose is given within, so these 100 gray peak doses are given within a second or less than a second, because especially if you have a moving target, you know, if you have an animal which will breathe, you can't deliver such fine structures of radiation, you know, such tiny geometries over more than a second, because it will completely the distribution will increase, the spatial distribution. You won't have the accuracy that you need.
Yeah.
And with flash, this means that you will deliver all the ionizations in an instant. So, and there's an oxygen effect. I don't know whether you're familiar with that. So oxygen is a sort of radiosensitizer. So in the presence of oxygen, radiation is 3 times as lethal as in the absence of oxygen. And in hypoxic tumors, you have very little oxygen, and that is one resistance mechanism for tumors, that they survive radiotherapy much better because of a higher level of hypoxia, because they have this bad vascularization and bad supply with oxygen. But with the flash effect, you will get this essentially in normal tissue. You will mimic the situation that you have in tumors in the normal tissue. So in normal tissue, you normally have a lot of oxygen compared to the tumor. But if you deliver all the radiation in a fraction of a second, then obviously the diffusion constants are too slow to allow for all the radicals to interact with oxygen. You will get depletion of all the available oxygen, and no oxygen can replenish that within the fraction of a second that all the radiation has been delivered. So there could be some contribution of such an oxygen effect, but I guess it's a combination of all of these.
Yeah, thank you. It's very interesting. Maybe one brief last question. Is it just normal oxygen that is the sanitizer, or some ionized form, excited?
You could use other forms of oxygen as well.
Form.
People have been looking even at prodrugs that could release some forms of oxygen or oxygen mimetics to sensitize tumors.
Okay. Thank you, Kai. And the name, in my name, and for everyone else, was a great talk, a great session, very insightful, very interesting.
Oh, thanks very much.
Everyone, thanks for being with us tonight. Kai, thank you again, and have a wonderful evening or day, wherever you are. Bye-bye. Thank you.